Hi, I'm Allie. I'm a fifth year PhD candidate in Dr. Bradley Peterson's lab. In the third year of my doctorate, I went to the University of Gothenburg to the Kristineberg Research Station, and I did a portion of my PhD with Dr. Eduardo Infantes. And Eduardo is an amazing scientist and a great person. He's currently an assistant professor at the University of Gothenburg and also a researcher at the Norwegian Institute for Water Research. Before he started his assistantship at the University of Gothenburg, I think he won the prize for best place to do a PhD. He was in Mallorca at the University of the Balearic Islands in Spain, working with Dr. Jorge Ternado Muñez and Dr. Alejandro Orfilia Forrester. And during his PhD, he also was at MIT, Woods Hole, the Netherlands Institute for Ecology, and the Horn Point Lab. And I was put in touch with Eduardo through Aaron Cox, who actually spoke here a few years ago. And together we have been really digging into these questions of eelgrass physiology and eelgrass restoration. And so with that, Eduardo, please tell us about what you've been up to.
Okay, well, I'm very pleased to be here and to be able to show you about the work that we are doing on eelgrass in the west coast of Sweden. So I'm going to start saying that I'm not working by myself. I'm part of a big team of environmental lawyers, environmental economists, and biologists. And we are working together. We call ourselves the SORRO team, which is a acronym of Sustainable Restoration. And we have been looking at the causes of eelgrass loss in Sweden. We have been looking at developing methods for large scale restoration and looking at the legal, ecological, and economical aspects of compensatory restoration. We have just started a Twitter SORRO group. So if you want to see what we are up to, you can just follow that direction. So yeah, our goal is to make national recommendation policies for eelgrass mitigation and compensation in Sweden. So for those of you who are not very familiar with eelgrass, most of you are, but just in case, I understand that there's a group with different backgrounds. So eelgrass is very important for the environment. It's stabilized the shoreline. It's very good at wave dampening and wave attenuation. It removes CO2 from the water and releases oxygen. It can also store carbon into the sediment and takes nutrients from the water column and from the roots as well. It improves the water quality, which makes it good for the environment and for the plants to grow even more. And very important, it's a very nice habitat for many marine species. And it's also a nursery for many fish, like here in Sweden we have cod, for example. It's like one of the most important commercial species. So in Sweden, eelgrass has been declining around 60% since the 80s. And the main causes of this is overfishing and eutrophication, which has caused a loss of more than 12,000 hectares of eelgrass. The causes is nutrients and nutrient inputs from eutrophication and overfishing. And probably most of you are familiar with what is eutrophication: increase of nutrients in the water, increase of algae, and this stop the light to the plants and then suffocate a little bit it stop the interchange of water between the plants and the water column. I'm going to be talking about several areas within the Swedish west coast, and especially the Gullmars fjord, where we have fairly good water quality and the plants are growing well, and the Kungälv area, which is we have been experiencing very high losses of eelgrass. For example, this picture is the Kungälv area, and all these red polygons are locations where we had eelgrass, and everything is gone right now. Well, almost everything is 95% lost, which is almost everything is gone. So we have been focusing on this area as an area for restoration. We are using the Gullmars area as a place where we are testing and developing restoration methods since the plants grow, but we are targeting Kungälv as a restoration place, and I will tell you about it. So this is how eutrophication looks when you have clean water here in Sweden. This is how a bay normally looks, where a species, Zostera marina, looks a bit like this. Probably you have Zostera marina there as well, so you know. So when you have a eutrophication event, we have a green massive event of filamentous algae on top of the canopies, and yeah, this is how it looks like. In addition to eutrophication, we have development of small ports and marinas. Sometimes it's little piers like these ones. These are very common within the fjords, where you have just a little pontoon with several boats. But this is a map where we have, sorry, it's in Swedish, but basically it's a map where we have marinas and these little developments. So even if it is a small impact of these little pontoons, when you have so many, this can lead to a very large impact on the eelgrass. So the situation, it has been that in the 1883, there was a big expedition and they mapped eelgrass and many other benthic habitats. But the interesting thing is that they saw eelgrass living down to 20 meters depth. Some years later, in the 1980s, there was another big survey and they found eelgrass at 10 meters depth, but not deeper than that. And the situation today is a bit different. We don't find any eelgrass deeper than 2 to 5 meters. So since 20 meters, now 10 meters, and then between 2 and 5 meters, what is going to happen next? How is going to be in the next 5, 10, 15 years? Are we going to lose all the eelgrass remaining, or are we going to be able to do something and change this situation and bring the seagrass back? To do that, we believe that restoration is the way forward, since plants are not coming by themselves, but there is a good restoration and bad restoration. As an example, there was a lady some years ago who tried to restore a paint, and this was the result. So as you agree, she didn't do a very good job there because she was lacking on painting skills. So it's important to develop very good restoration methods if you want to do a good restoration work. Otherwise, you can make a mess. So in Sweden, over the last 10 years, we have been developing restoration methods. I'm going to show you two. One is using transplants and another is using seeds. So basically, transplantation of eelgrass, you just pick up plants from one location, plant them in another bay, hopefully close in distance. So closest bed, you use it to transplant, to pick up the donor. And we have been testing this in many areas around the west coast of Sweden. And basically when we plant 12 shoots, if things go well and it's the right location, after 1 year, we can have a massive growth, around 200 plants per square meter, which is quite good. We did this with small plots, but since in some areas it's working, we have been increasing the sizes. So this is a plot in 2015, 10 by 10 meters. And we can see the progression over the time. I took these photos with a drone because, as you know, the water here is quite cold. So we thought that just sending a drone could be a bit easier than just jumping in the water. I'm Spanish, so yeah, that's how it is. So, but in doing this, we have been developing methods using drones, but I will tell you about that later. Now, here we can see that the plants have kind of survived over 1 year and the density, it increased. from the planting, which is 16 shoots per square meter, to more than 95 shoots per square meter. So we planted this in 2015, and today we still have these plots around. In this photograph here, we can see these are the squares I was showing you before. And in 2019, we planted even larger squares, which you can see here. So this is just after planting. And this is some months after. And these are 20 by 20 meter plots. And with this, we tested different patterns to see what is the best configurations and to estimate the cost. We're looking at biodiversity returns after restoration and so on. So these are started with small plots, then 10 by 10 meters. This is 20 by 20. Last summer, we have been planting a larger plots, and I will show you this is in a location in the Coster Habitat National Park. This is close to the Atmosphere. And in this location, there was some mapping done in the 84 and the 80s, where you see these blue dots. This was all full of eelgrass. Now everything has disappeared. Even these other spots where, well, it was mapped in 2008. Right now, the only remaining is these green areas right here. So in this big bay where eelgrass is absent right now, we have been planting larger plots. So this is more than 120 meters long with 40. And yeah, using the 1 square meter checkerboard pattern. To see the scale, you can see here 4 divers in the water doing the planting, and this is the diver's boat. So yeah, we have been increasing the scale of the plantings. I want to tell you also that we have been testing with seeds, and basically the idea of seeds is to collect the flower shoots, which are up here. You collect them, you put them in tanks, and when the seeds ripe and mature, they drop to the bottom of the tanks where you can collect them. You store them in tanks, and then you place them in the field. And if things go well, those seeds will become a seedling, and later on a patch or a meadow. But after so many studies, we have seen that here in our area, it was a very low survival of seeds and seedlings, around from 0 to 10%. And if we are thinking about large-scale restoration, perhaps transplants have shown to be more reliable than using seeds. So all the techniques that we have in seeds, because yeah, it's just basically cheaper to do it and more reliable. But the process is you pick up these reproductive shoots, you put them in tanks with some aerated flow, and then you pick them from the tanks, you store them in the lab in climate control rooms, so with flow through, and then we place them in the field. After planting in the field, you can see the seedlings coming from the seeds, and after time, you have these little patches happening. So all these methodologies and all these restoration techniques, we have publications, but we have developed some manuals for management here in Sweden. So these are really big manuals that are only in Swedish at the moment, but in a few months they're going to be released in English. So I can let you know if you're interested to have a look at what we have been developing. In addition, we make some video tutorials, video guidelines, to show how actually we do the test plantings and the monitoring and so on. You can check all this in my website. We have them in Swedish, but we have also in English as well. Okay, so we see that when we are planting in some areas like the Atmosphere, the plants are doing well, the restoration works. But in an area like the area of Kungälv, when we plant, everything dies. This is very challenging because, yeah, restoring is quite difficult. And there are some feedbacks that are preventing seagrass growth. So in the next slides, I'm gonna show you a little bit about 3 feedbacks that we have identified in our area. One is sediment resuspension. We have also drifting algae and predation. So I'm gonna go through these 3 different feedbacks. To start off, I'm gonna talk to you about sediment resuspension. And basically when you have seagrass, you have a reduction in the flow because the canopy reduces the flow and the waves. So the sediment is more stable in the bottom and you end up having better, quite good water quality, which leads into more seagrass growth, right? So we have this called a feedback loop, positive feedback loop. When there is a disturbance in the meadow because of eutrophication or anchoring or these small piers, these marinas, there's less canopy to reduce the flow and you have more sediment resuspension. If you keep increasing the disturbance, then you can reach a tipping point where everything is a bare sediment, so you don't have any eelgrass there anymore. So if you don't have eelgrass, you end up with bare sediment and there is higher flow because you don't have the canopy there anymore, which leads to high sediment resuspension and poor water quality. So in these feedback loops, plants are not growing there because the bad water quality and bad light. So we have this scenario in the area of Kungälv. And I will show you some examples. This is a site where it used to be covered with eelgrass, all this area, but now everything is gone. And with a little bit of wind, we have a massive resuspension plume of turbidity. And this is happening there, just right, with a little sea breeze. It's not a stormy condition. There is no white capping or anything. It's just quite mild sea breeze. And the turbidity is not coming from a river or anywhere else. It's just happening there in an interval of several hours. I took these photos with the drone, so it was still okay to fly there. Not so windy. You can see very clearly the plumes. There you can see from the boat, these little plumes developing. And this is caused by glacial mud, which is very fine particles that get resuspended with a little bit of orbital motion and makes the water very turbid. We believe that before, when you have the canopies, they were protecting this sediment and it was not being resuspended, but now that the canopy is gone, then it becomes resuspended. To prove this, I can show you an example. This is another bay in the area of Kungälv where we had all these massive losses, and this is also a fjord, part of a fjord that it was covered with eelgrass, but now everything has disappeared. And during this is a big storm event with winds of 30 knots, so it's not a sea breeze, it's a bit higher. We can see that when we had this really strong wind event, then the light in the bottom, we measured with some loggers, then it went down to 0 over several days. And during this time, the turbidity, it really increased, it really spiked over that. So we can say that this is caused by sediment resuspension and this turbidity, which is happening locally there in this base. Another feedback and tipping point that I will show you here is a drifting algae. This white polygon, it shows a meadow that disappeared, so it was more than 200 hectares. Right now you see some darker area in the center, and this is not eelgrass, this is drifting algae, which is Fucus, mainly Fucus. I think this is Fucus serratus, and it's just moving around the bottom. And when we plant, it kind of goes on top and erodes and kills the little plants. So this is preventing restoration in this base. So when you have plants, then you have good water conditions, there's plant growth, similar to the other feedback loop. When you lose the eelgrass, then you have this macroalgae coming into your bay, and this drift algae will cover the whole place, and it will become low water quality with the movement, will be resuspension, and so on. So this is what's happening as well. And another example is of a feedback that is preventing restoration is crabs. This is Carcinus, which is a common species here. And I don't know if you're gonna see this time-lapse video, but I placed a GoPro and I just capture how Carcinus is eating a shoot. But we also did we'll leave it there running, so maybe you can see something. We did a study where we measure how were seeds, how were the crabs eating, who was eating the seeds and the shoots. So we saw that Carcinus, the short crabs, they were big predators of seeds. There was also Hermicras and sea urchins that they were eating, but it was mainly the crabs. And the story with crabs is a bit more complicated and quite relevant because cod has been overfished over the last decades. Here in this graph, you can see the decline of cod stocks over the last decade. And since cod are eating crabs and other small fish species, these species have increased as well. So there's a relationship there. So since cod are eating crabs, and we know that crabs are eating shoots, this is causing a feedback loop here, because if we lose the habitat for the juvenile, then we will end up also having less cod. In addition to this, if we add eutrophication, which is the addition of nutrients into the water, we have an increase in filamentous algae, as I showed you earlier in those photographs. And there is evidence that these filamentous algae are really good places for juvenile crabs to recruit there. So the more filamentous algae, the more crabs, more predation on the plants, less habitat for the juveniles. So this is actually it's a double feedback loop that has having effect with the crabs. Okay, so these are the three feedback loops I wanted to show you. Now, how can we break all these feedbacks that are preventing recovery? I'm gonna focus in one of them, which is quite exciting to me. I have a background in physical oceanography and relations interdisciplinary with ecology. So I like very much hydrodynamics. So this is one of the bays in the area of Kungälv, where we used to have a big eelgrass meadow, and everything has disappeared, and we have these plumes that like the one I showed you before. When there is a bit of a wind event, you have these plumes. I also took this photo with the drone, and we had several questions. How can we break these feedbacks? So if we add a layer of sand to cover this mud, are we going to improve the water quality? But where should we put the layer of sand, and how big should be the area? And we had many questions. So over the last year, we have been working on this, and we developed first a hydrodynamic model, and we use this DHI Mic 3 to look to build from a large scale of the whole coast. We had our little bay here, and this is the place where we are really working. So this is this bay here, but we're assessing the large scale conditions to really model well the local scales. So the model simulates wind and sediment resuspension with turbidity, and yeah, we also carry some field observations, and the model matches quite okay compared to the field observations. So yeah, we can say that it's working. And we did many tests, and I'm not going to bore you with all the different simulations we did, but we tested different sizes of polygons and locations depending on depth and so on. We end up with one case scenario, which is 1 hectare. If we place this square in this position in the bay, we could reduce a little bit the turbidity enough for the plants to grow there. So yeah, this is what the output was. And today, as we speak, there is we are actually sand capping this 1 hectare polygon. We are not doing, we hire a company, we are professionals, and they are dumping this 10 centimeter layer of sand on this. It's a bit messy right now, but we are looking, also we are monitoring, here is our boat. We are looking at the turbidity and so on to make sure that this is under good levels. But the interesting thing will be after this is done, when we will be planting in June and July 1 hectare of shoots with the same densities as I showed you before. In this photo, you can see, well, it's the mess that they are creating because it's very fine, between 0 to 8 millimeters grain sizes. But here you can see the first layer of the sand cap happening there. They started yesterday, so it will take about 3 weeks to finish the job. But maybe I can show you some of this more in the future. Okay, other tools we have been using to assess this resuspension and turbidity is a flume that I have developed at Kristineberg here in the marine station. And in this flume, here you can see the sand cap that we tested, and to see if with the flow velocity this is what's going to remain in the place, or it's going to be washed out after the first storm. So for this flume that I built a couple of years ago, we can generate waves in this direction, and then there is a current motor here that pushes the water underneath, and we have a current. There is wave gauges and flow meter and so on. This is how it looks. I don't know if you can see well the video with the waves in the flume, but yeah, it's a little short video. I have also developed some wave mesocosms. This is also at Kristineberg, and with these ones we can test growth of plants under different wave exposition. And this is 4 tanks with a wave maker. This is small, and we can generate replicated experiments over long periods of times with a simulate the bottom shear stress that we find in the field in the flume. I actually just published a paper with this facility, which I hope it will be out next week. It's now in press. And we have filmed a little video. This on Monday, I was filming a little video to make a video abstract. So once I have it, I will share it with you. That was quite fun. At this point, you might see that I like flumes. And this is another flume we use in the field to see sediment resuspension and turbidity. And this is a field flume. Basically, it's a motor that we place in the inside of this chamber and generates a unidirectional flow in this direction. And here we have a turbidity logger and the seagrass. This is on the edge. So this is eelgrass here, and this is the sandy area. And we have detailed cameras and a flow, an ADB flow meter here. All the motor and everything is driven by a battery, and we are measuring things with a computer in the boat. So instead of bringing the samples into the lab, we are bringing the lab into the field, and that's quite cool. Yeah, this is a short video of how it is. In addition, to look at the orbital motion, this is another flume that we developed with some colleagues from the Netherlands, from the NEOS. And this is a wave flume that basically has 2 pedals, and they move back and forth, and they generate a shear stress in the bottom. So we can look at resuspension and turbidity in a different way. Here you can see the acoustic Doppler that is inside, measuring the flow velocities. So yes, so this is a bit the stories about the feedbacks. And I want to finish a bit the talk showing you sharing with you some images about work that we have been using with the drones. Perhaps you are using similar techniques there. So here in Sweden, we are developing methods for seagrass and coastal monitoring, habitat monitoring. And we are evaluating this using satellite images and drones combined with ground surveys using aquascopes or drop video cameras and GPS and so on. So, but perhaps I will tell you about this in another talk, because that's quite a long topic. But we find that drones are very useful for doing coastal studies. For example, what I showed you earlier on this restoration and monitoring. Also very useful when we are looking at the factors preventing restoration, like these feedbacks. And we have been also for fun looking at how drones can be useful for marine mammals, and how we can measure the puppies of seals and measuring the body mass index using the drones. And lately, also related to seagrass, in Mozambique, I have a PhD student who is looking at how these are the dugong feeding trails. You see all these marks here. They are on top of an eelgrass bed. So yeah, we can measure and calculate all these feeding tracks for the dugongs using the drones, because they are over a very large area. And we have also looked with the drone at mating behavior in Thailand for the dugongs. And we also just published this. So in summary, eelgrass in restoration in Sweden is possible, but in some areas we have feedbacks that make restoration challenging. In some locations, like in the area of Kungälv, as I showed you, especially with sediment resuspension, drift macroalgae, and crabs, who are really a problem. With economists, we have estimated a cost of around 0.1 to €500,000 to restore 1 hectare. So instead of thinking about restoration, now we think that it's better to conserve the eelgrass beds instead of restoration, but if there is no other choice, that's around the cost. The cost, it will be 0.1 if you don't include any other measure as sand capping. If you add other measures, it will be more expensive, of course. And at this point, we are testing implementation of large-scale restoration methods here in Sweden. So with this, I just want to say thank you very much. This is from Kristineberg. Here's our marine station in a summer day. But if you come on a day like today in winter, it looks a little bit more like this. So thank you very much. If you have some detailed questions, here's my email. There's also some more videos and research articles and everything in my website. You can just go there and download. Thank you very much. If you have any questions, I'll be happy to answer now.
Oh, wait, someone has a
Very
question.
much. Very interesting. I think people can either put a question in the chat box or just speak up.
I wanted to give others a chance, but since no one's jumping at, I have some questions. Eduardo, so when you do your plantings where you mentioned the crab predation. Do you have issues with crabs just pulling your shoots out if it's not at a high enough density? I'm just curious about the numbers you're putting and whether or not you see a density effect that kind of overcomes some of these animal removement.
Yes, we did a test and checking the different densities, because that's a very relevant question. And we saw that in some places we tested 4, 8, 16 shoots per square meter. And depending on the location, even with higher, what we think is higher, 16 shoots per square meter, and you can put more, but then restoration becomes really expensive. So it has to be a threshold there. If you put 100 shoots per square meter, maybe it will survive, but it will be unrealistic if you want to do restoration like that. So yeah, it's very much depend. Some bays there are many crabs, other bays for some reason there are not so many. A way around maybe is to do some fishing and put some cages and grab the crabs, dump it in another bay, or I don't know. So
a lot of effort here has been done in the Peconics, and we've got spider crabs, and they just seem to go towards anything that's been put in to just tear it up, which is very frustrating after the hours you spend doing the work.
Yeah.
And I appreciate your showing us your drone stuff. We're very excited about that as a metric of really evaluating the outcomes of your restoration, which I think is really, really important.
Yes, it's a very useful tool, and I think we are just at the beginning of it. So yeah, it's great.
Hi, Eduardo. I have a question for you.
Hey, Erin. Nice to see
I
you.
really enjoyed seeing you again and hearing your talk. For your restoration work, have you guys noticed anything like more success with different maybe donor beds or sources that do better than other sources? Because like a lot of interest is happening right now about genetic diversity, and could you increase the genetic diversity of the bed, and that would allow it to restore at a greater rate or restore and be more resilient to stress. And so I'm just wondering if you guys had any sort of tried that or any insight into source populations and if they matter.
Yeah, so with the we just also scratched the surface on that. We have in the team Marlena Janke, who is working a little bit with the connectivity and this genetic part. We have been, but we haven't seen any clear trend in this. We have been doing some more basic phenology type of transplantations, where we transplant from deeper water to shallow water, and from an exposed place into a sheltered place, because maybe the plants, depending on the restoration place, might be adapted to more exposed or more shelter. So we have been doing all sorts of tests in that way. And basically, if you plant a big shoot from a sheltered place or from deep water into a shallow, they will not make it. They will be broken or they will be dragged. Apparently, in sheltered places, they are more fragile. They break easier than if it is an exposed place where they bend much more. But if you transplant from an exposed place into more shelter, it seems to be working a little bit better. Or from shallow into the deeper, which in the shallow, they tend to be a bit smaller, and in the deeper are longer. So they do better, these ones. So that's what we have been finding out.
I don't want to jump the gun, but since no one else asked, I was watching, your images of your restoration plots over time, which were really interesting. And I was wondering, do you observe or follow metrics of diversity or utilization by the fauna on that same scale where you're taking your photos from?
We are not doing fauna so much at the moment. We just started a little bit with some colleagues from Finland, who they are coming to our restoration plots and taking the samples of infauna and epifauna and looking into that. So, but we have been more focused on the methods, on the restoration techniques. But these plots that will have the different patterns, those ones we are starting to look into yearly variations in the infauna depending on the patch size, and that's quite exciting. So we have finished all the sample analysis. At some point, we'll have something ready to share, but at the moment, I don't know.
Well, my mind just goes to the cost and the effort and expense. You always need to justify and being able to look at the species diversity enhancement or the increase in faunal utilization makes that argument for the dollars cost.
Yeah, absolutely. Absolutely. I agree.
Niels, do you have a question?
Yes. Hey, Eduardo. Thank you very much for a nice presentation with beautiful pictures. Really nice.
Thank you. Thank you
So,
very much.
sorry, I was a little late, and maybe you mentioned that in the beginning, but looking at Sweden and looking at the landscape there, it's so, well, it's not very densely populated. And I wonder, what are the major sources of eutrophication at your site? And is it getting worse or better? And what is done to, well, make it less bad? So I'm just surprised. Does it all come from Denmark and from Germany, or is it like locally produced?
I don't know who we can blame for that, but I think it's more of a Baltic problem. There's many countries around the Baltic, and there are some bigger polluters than others. It's true that there is more regulations and restrictions in the dumping of nutrients in the water. There's better treatment works now. So it seems like the pollutants or the nutrients are not increasing. It's kind of, yeah, at least it's stable. It's slightly decreasing the amount of nutrients, but even with this, the plants are not coming back. So, yeah.
Thank you.
Well, since no one's going, you know, I think that your point that you raise about these feedbacks and tipping points is really critical in the concept of restoration, right? I mean, we see lots of times where people say, we used to have grass here, and so we'll put effort in putting grass here, but the system has fundamentally changed. And I think that it's really important to link this change and these ideas of tipping points to restoration. In fact, you know, in my mindset and worldview of what we are doing here, I'm really looking towards really ecosystem manipulation more than restoration.
Yes, I agree. It's tricky, and yeah, many things happening, and these problems could be, I mean, it depends. If it is like a kind of like a global large scale problem, maybe it's that more difficult to fix. Climate change, that in your bay you cannot really fix. Temperature rise, but other local problems, perhaps it's easier to find solutions. Maybe it's easier or less costly. Others maybe might be more expensive, but I think what the approach has been to start small and then increase in scale little by little, finding if you jump into big scale, you have many chances to fail, and then you will lose kind of credibility on your science. So it's better to do little steps at a time, make sure that every step is solid, and then restoration can surprise you. You can even go large and it might fail, but I think it's important to go little by little up in scale.
So I have a question. This is Josie. In some of these embayments where you've lost seagrass, do you have any problem in thinking about reseeding with microalgal or cyanobacterial blooms, or you know, is that not a problem?
We thought so. That's quite relevant. We thought so that it could be the turbidity, that it was not resuspension. So when we measured turbidity, I didn't show you the data because I thought it was perhaps more clean, but we measured chlorophyll A, and we saw that these turbidity events, it was not caused by algae blooms. So the turbidity was mainly driven by resuspension. The chlorophyll A was pretty flat during this resuspension and low light events. So they could be happening, but maybe this is happening more in deeper part of the fjord, but not so much in the very shallow. And seagrass is very shallow, between 1 meter to 3 meters. So maybe deeper parts is more relevant. I don't know.
else here? Any other questions, people, students?
So I want to talk to you about seeds for a second.
Yeah, do that.
So we get about 10% survival on seeds. And I'm wondering, our system is so different than your system. I take one step away from where I am and my odds of finding a clone, are like 5%. So it's a very different kind of matrix across the landscape. And what you were talking about moving, shoots from shallow to deep or deep to, when those seeds are growing and those genes are turning on or off, depending on what kind of, location that they are at, to me is like, that's kind of what I want to get. I want to be able to use that. But, the issue of like having a matrix for the seedlings to be able to survive in while they're growing is not trivial, especially when you're talking about things like hydrodynamic stress, like you are talking about. And do you have other seagrass species than rupia there? Do you have any rupia other than? No, so you can't do within a matrix of another species.
No, we only have a bit of novelty and it's super shallow, so and very rare, so we cannot really use that one. And rupia is very much into very fresh water and also between at 1 meter depth, so it's they don't really overlap. Yeah, maybe we need to go to the tropics to do that.
Yeah. I thought that Niels was going to talk to you about the question, look, when you had those seed work that you had done, all I saw were lugworm mounds. And I thought for sure that's what Niels was going to talk to you about. And then I got convinced that Niels was going to talk about his envy of your in situ flume, which I also have as well. Some pretty cool ways of going after these questions in the field. I applaud you for that.
Yes. Well, if you want to come over and try your questions here, we have some gadgets that we can use to try to answer some of the questions. Yeah, there is in the very shallow, there is actually a lot of Arenicola marina. It stops at around 2 meters depth, but the very shallow, that photo that I took of the shoots, it was one of the shallow plots that we had. So yeah, it's plenty of Arenicola. And yeah, I'm also a big fan of flumes.
Hey Eduardo, nice to see you.
Hey Claudia.
Such a long time. I had a question about the seed-based restoration. Obviously, Ali and I did our seed experiment in Sweden with you. And I wondered, what is it specifically about seeds that means that the success rate is so low? I know you mentioned predation by crabs, but obviously if you're doing it in a lab and there are no crabs coming in to eat your seeds, what is it that is so difficult about rearing seagrass from seeds?
Yes, I just put the slide on. I don't know if you can see. The difference with the seeds is that there are many little steps involved in the restoration. So when you pick up one plant and you plant in another, you just, in one day, you can do it. So you can just pick and plant. But when you are doing seeds, then you need to pick up the flower shoots, bring it into the lab, have them in a nice condition so they can finish developing and producing the mature seeds. Then you need to collect them, store them, bring them into the field, find a good way to plant it in the field. And so also the time extent is much longer. So basically if we will plant, we can go in the same day, pick up the shoot and plant it. And that's it, it's already planted with the root system and the rhizomes. So we could see in a few months if it grows or not. Or we can come the next day or the next week and we see if it is doing well or not, or if it is there or not. And we know where we planted. With seeds, it's more tricky because the time span since you collect the reproductive shoot or the flower shoot until you have the seedling there is much longer. So when you put the seeds in the field, I don't know if you can find it again. Maybe if you cover it, so you don't really know if, what happened. We have been using cages to really make sure that the seeds were in the same place, for example, and then we could find the seeds to test predation. But other than that, it's just more complicated, more steps, so many unknowns. But I think it's worth developing more techniques on seeds. I think that's a good thing. Maybe it could be cheaper in the long term, but at this point, we found that it was not very practical. That if you need to recommend to a company, we need to restore 1 hectare. If you put all these seeds and nothing happened, then it's a very, very big failure. So we found that with the transplanting, the chances of success is higher. Everything can fail because it's very unpredictable to work in the sea. I mean, in a field, you can put a fence, you can plow the field, you can add nutrients, you can add water, but at sea, anything can happen from a boat to many things. So yes, we found it's a bit more tricky at this point.
Okay, so transplantation is the way forward for sure.
For now, with
So
far, that's what we see. But maybe in the future, I encourage more seed work. But for now we are using transplants.
Yeah. Well, thank you. Also love the drone pictures as always.
Thank you.
I have a question actually on that note. So have you done any studies where you have these like really, so you have these like beautiful plots that have been, that have increased in density over time, right? And then have you gone back to those plots and maybe tried to do seedling experiments, either using like burlap bags or whatever, like, but just using the seeds and then sort of following that through time and looking at the genetic diversity to see if you can actually, so if you have a plot of eelgrass that you've restored using your transplant methodology, which is like very exciting and very like visually very cool to see that through time. I'm sure Brad is gonna probably try to make our lab do that this summer. But again, because like if you have this, this patch of grass that's pretty robustly established and it's been there through time, to then go back to try to add in the genetic diversity to sort of try to bolster that. And I mean, I feel like this would probably be years, right? Like a long-term commitment, but maybe the two things really need to happen in parallel, or I don't know.
Yeah, well, in the plots that we have been restoring, we see that after time we get flower shoots. So that means that we could collect those seeds and use them in the future for something. We haven't done it because we have been more interested on developing the planting. But now that we have all these restored areas, this open the possibilities for new experiment. To me, I see it like a new lab facility to have a restored place where you can look at biodiversity, you can look at stored carbon in the sediment, you can do many things because we have a date where it started, which in a meadow, you don't have that. Now we say, okay, we know that in the June 2016, we planted and we know the conditions of the sediment at that date and so on. So if you have some cool ideas, I'm happy to hear. You're welcome to come over. I wish I was there and I could join some field activities with you this summer. Yeah, actually, it would have been nice to be there and give the seminar in presence.
Yeah.
So.
Yeah. Okay. Are there any more questions to the?
I have a question.
Okay.
Oh, hi. Cool. So I'm a new master's student in Brad's lab, and I was really interested in your sediment transplantation. And I had a question about, now that the rhizosphere and incorporating microbiomes into seagrass ecology has become new and really exciting. How are you concerned with that at all with the sand transplantation? Because we see like with beach transplantation, it can affect the beach communities. Are you concerned about that with the seagrass?
The beach communities? What do you mean?
Well, like so if, like for instance, I know of if a beach is changing composition because they try to add more sand to it because the beach is eroding away, it could affect, beaching sea turtles don't return there anymore because they don't recognize the beach community anymore, and the composition of the sand has changed, so the community itself changes with the sand.
Yeah, I mean, adding sand is a big change into one bay. So we have looked first before we thought about starting adding sand. We did an impact study, a small impact study, to see if we were killing something, because there's no point of restoring if you are going to destroy something that is there already. But in this glacial mud, it's so muddy and so packed that there is nothing really living there. Or well, it is, but it's very low number of nematodes and so on. So when we added the sand, we saw that actually the coarse grain brought more animals and so on. So, and we are going to be monitoring this biodiversity over time in this new plot. So seems that adding sand in this base could be a good thing. And especially if we manage to bring the seagrass back, it will increase biodiversity, hopefully. Let's see, we'll find out.
We'll be very excited to hear about this, because people around here are talking about using sand capping over the organic muds. And so very, although different bottom sediments, it'll be interesting to see how your sand capping pans out for you. Yeah,
we are very curious as well. But actually, sand capping is not a new thing. For example, we are ecologists. We are not used to doing this bringing cranes into the field. We are more used to buckets, perhaps, but not like a cranes with tons of sand. But this is in industry. This seems to be quite common to ameliorate the polluted sites, to cover them with clay or sand. So apparently, there's companies that they are specializing in doing this type of activities. So maybe we need to join a bit more interdisciplinary and bring engineers into our field and work with them.
So Eduardo, I have to ask you, because I desperately need to know how you so when I was there, you were looking at identifying eelgrasses from your drones, and you had like you had like 4 different drones, and you're working on that project, and you're identifying like the eelgrass versus like the macroalgae mats and all these different things, and it was like so awesome. And like, how is that project going?
Yeah, that's moving forward very, very well. That's I didn't want to enter into detail to not be too long into the but yeah, this we have been doing this for several years. So basically we have been flying the drone over different bays and comparing with satellite images, and in order to monitor eelgrass vegetation. Here in Sweden is now it's going to become by law with the Water Framework Directive to monitor habitats, and every country is going to need to monitor this, otherwise they are fined. So it seems to be working well. We still more development, but with the drones you cannot cover a whole coast, which the Swedish coast is very long and complicated with all these fjords. So a combination of drone plus satellite images seems to be the best. Because with just one image from the satellite, you can cover half of the coast, which is amazing. We're using Sentinel-2. The problem with Sentinel-2 is that the resolution is not so amazing. Well, better than many years ago, but it's 10 by 10 meters. With the drone, you can have a much higher resolution, to 5 centimeters. So you can see better. So you can see in bays where you have more fragmentation, for example, you cannot capture that with a satellite, but with the drone, you can really see more patterns and so on. So I think a combination of both satellites plus drone is very good, especially if you want to monitor. So the idea is to calibrate some of the satellite data with the drone. And perhaps it's difficult to monitor the whole country, but specific bays you can monitor over certain, and so those bays we can do with the drone and see how they change, but then get the aerial cover of eelgrass or vegetation at the national level using the satellites. So it's going in this direction more or less. But yeah, the things are working.
That's so cool. I would like to use your in situ drone. I'm sorry, your flumes, please. I will talk to you about this later. That's awesome. That is so cool. You were starting that when I was there, and so you actually have been able to ground truth everything and troubleshoot everything, and it's working now.
Yeah. I mean, we are still working on it, so it's not perfect yet, but yeah, it's hopefully will be it's a work in progress. Yeah, but it's kind of working.
That's awesome. Congratulations on that. That's so cool. Okay, is there any more questions from the group at large? Everyone?
I had a quick question. I was just curious so with these really large scale restoration projects that involve taking from a donor community, how conscious of you are potential impacts on those like donor communities, and are you like monitoring their recovery from once you take the transplants?
Yes, first of all, Jessica, I like where you are sitting right now. That's amazing.
We had our first 60-degree day, so I have to be outside.
Yeah, in the hammock. Okay, that's really cool. So I forgot your question.
So it just has to do with,
I was just focused on the hammock.
From the donor communities, when you're taking these transplants, are you monitoring their recovery, and are you ever concerned about taking too much, and how do you sort of mitigate that?
Yes, that's very key. So one of the first things when we started the restoration test experiments from like the very beginning, it was that are we really causing an impact on the donor beds, because otherwise there is no point of trying to restore if you are killing something else. So yeah, we saw that actually decreasing the number of shoots into a like very dense meadow where you are picking. Of course, you want to pick in a dense place or like a good place, not like a very fragmented. So this actually increase the number of shoots. It's like if you are kind of like land mower your land, your grass, that it becomes a bit more green and more dense. So it seems to play that effect. So not big impact, at least when we were measuring. We followed those donor beds over a long time. So when we are developing the techniques and these handbooks and videos, we added this recommendation, and also as part of the monitoring method. So if you are restoring, you need to monitor your transplanted area, but also monitor your donor location to make sure that it's also fine. So because you want to recommend this to companies and so on, but that part is really key. Yeah, so it's included.
Yeah, thank you. I'm glad that you're incorporating that into these recommendations, like to companies.
Yeah, I mean, we are pro conservation and pro nature, so we don't want to destroy something to fix another problem. So yeah, that's very important.
So Eduardo, what time is it where you are right now?
It's around 7. It's about beer time now.
Yeah, time for a pint. That's exactly what I was thinking. Eduardo, thank you so much for taking the time to give a seminar here. We really appreciate you doing this in your evening over there.
It's a pleasure. It's a pleasure, and that's how it is. The world is round, so so it's a pleasure to be here, and I'm very happy to really be able to share all the work we have been doing. Hopefully, I hope something is interesting or relevant, or like you can use it, and hopefully in the future we have a different world and we can travel more. I will really like to come to see you there.
Yay, we want you to come here.
Yes,
it's so fun. Okay, thanks so much, Eduardo. It was such a pleasure.
Yes, a pleasure.
Bye.
Bye.